Yes. Good evening, Harsh. Yeah, Ashish. Yes. Good evening, everyone. Kiran. Yes. Okay, perfect. RS Creation, Sulphase, Anshul. Good evening. Good evening, beta. So today we are going to start with the biochemistry of carbohydrates, part 3. So before starting, just give me a thumbs up in the chat box that the audio and visuals are perfectly fine, so that I can start with the yeah. I can start with today's topic. And moreover, as I have like promised to each and everyone over here that I will be discussing, yeah, the questions, okay, MCQ questions today. So I will be discussing the MCQ questions along with the one-liners. So you have to like pay attention to all the content that we will discuss today. Yes. So Jay Shri, Chirag, Shraddha, Kiran. Okay, perfect, perfect. Yes, congratulations. Yes, it's a pride moment that India has stepped on the moon. Yes. So with that note, just let's begin with our class. No, we are going to start with the oxidation of sugars or simply the carbohydrates. Simply the carbohydrates, beta. Whatever is required, beta, we will only study that. We don't have to study chemistry over here. So we are studying biochemistry, and whatever relevant points will be there, I will be discussing that. Right? Whatever relevant points will be there, I will be discussing that. Right? So yes, now oxidation of sugars, or I can simply say carbohydrates. So now a basic concept that you have to remember, beta. A basic concept that you have to remember. Okay, a basic concept that you have to remember. You already know the carbohydrates are aldehyde. Okay, they are aldehyde. Yes, they are aldehyde. Now, when I say aldehyde, beta, you already know it is CHO group. Yes, CHO group, and they are keto derivatives. Okay, they are keto derivatives, that is C double bond O. So remember, beta, whenever like these sugars are oxidized, whenever they are oxidized, so they will form acid. Whenever they are oxidized, they will form acid. Acid will be formed. Okay, acid will be formed. Okay, so yes, so in simple words, whenever there is oxidation of sugar, okay, whenever there is oxidation of sugar, what will be formed? Acid will be formed. Acid will be formed. Acid will be formed. चलो ये इतना बेसिक क्लियर हो गया सब लोगों को, इतनी चीज सबको समझ में आ गई. But from my end, the things are very much good. So please don't try to disturb the class. Please check at your end also. Right? And yeah, I hope the screen is like perfectly fine, beta. Just give me a thumbs up that the screen, audio, visuals are perfectly fine. Yes, Alfez, Harsh, Ashish. बेटा प्लीज चेक एट योर एंड, अपनी क्वालिटी बढ़ा लो बेटा यूट्यूब में, ठीक है ना. सो या डोंट ट्राई एवरी टाइम टू नो डिस्टर्ब. यस, even whenever the sugar is reduced, okay, or whenever reduction takes place of sugar or carbohydrates, alcohol is formed. Alcohol is formed. So that is like takeaway. That is like the takeaway message beta from here. Whenever sugar is oxidized, acid is formed, and whenever sugar is reduced, alcohol is formed. Alcohol is formed. Right? Now, before wasting much more time, before like, okay, without wasting much more time, beta, you already know that we have a structure that is called as glucose. Glucose. You already know glucose is actually a 6 carbon compound, right? It is a 6 carbon compound. I will not draw the entire structure, beta. I will just try to focus because we have already learned the structure of glucose, right? So you already know that the glucose is having 6 carbons, okay? 6 carbons, and the functional group beta, it is present at the first carbon, that is the CHO group, right? Yes. Now, whenever the oxidation will take place, whenever oxidation will take place, beta, whenever oxidation will take place, what will happen? This aldehyde group will convert into acid. Okay, oxidation, or I can simply say the C1 will be oxidized to form acid, because C1, it is the carbon 1 where the functional group is there, right? So what will happen? Acid will be formed. So yes, it will be COOH, okay, at the first carbon, whereas all the carbons will remain the same. All the carbons will remain the same. The first place, okay, it will be acid. And yes, so remember beta, it is like, okay, remember, it is mild oxidation. Now I'm taking mild oxidation of glucose. Can anyone tell me the name of this compound beta in the chat box? Can anyone tell me the name of this compound in the chat box? I want everyone to answer this question. Can anyone tell me the name of this compound in the chat box? Yes, beta. Glucose से बनता है gluconic acid. Again, be very clear with the names. It is gluconic acid. मैंने पहले भी बताया था. Glucose is a sugar. Glucose is a sugar, or it is a carbohydrate. Okay, it is a carbohydrate. Or yes, Sanjay, very good beta. Kriti, very good. Harsh, very good beta. Very good beta. Kiran, very good. So you already know the sugar or carbohydrate, the monosaccharide, that glucose, when it is oxidized, mild oxidation if takes place, it will form gluconic acid. Now beta, clinical significance is very, very important over here. Clinical significance is important. Yes, this gluconic acid, okay, when I say clinical significance beta, I'm just drawing this stethoscope. Okay, I'm just trying to draw the stethoscope. So remember, it is a sign of clinical significance. Okay, clinical significance. You already know we are dealing with medical biochemistry beta. So we don't have to follow only reactions. We have to see what is the use of this particular reaction. We have to see what is the use of this particular reaction. So beta, it is used for, like, it is an, okay, enzymatic method of blood glucose levels. Blood glucose levels. Okay, blood glucose. Okay, so yes. It is also called as the glucose oxidase method. Okay, estimation of blood glucose. Okay, by this it is also called as glucose oxidase method. And remember beta, that gluconic acid, it is also known as, can anyone tell me the other name for this gluconic acid? Yes, can anyone tell me the other name for this gluconic acid? Prince Panchal, good evening beta, good evening, welcome to the session over here. Welcome to the session. Yes, gluconic acid, it is also known as aldonic acid. It is also known as aldonic acid. Okay. So yes, we will be learning all these procedures. Okay, after some yeah, in some classes, right? So remember, the gluconic acid, it is used for enzymatic method of blood glucose. Okay. So yes. Now coming on to the next part, beta. Now it is like yeah, it is like the mild oxidation which is taking place at C1, okay, the case 1. Now taking case 2 beta, this is case number 1. This is case number 1. Now case number 2 beta, case number 2, under mild oxidation, under mild oxidation, remember, if the sixth carbon atom is oxidized, if the sixth carbon atom is oxidized, if the sixth carbon atom is oxidized. Now you already know, मेरा blood glucose levels के लिए होता है test, ठीक है, to see the blood glucose levels. Okay, now coming on to the sixth carbon beta. Sixth carbon, you already know we have CH2OH. Okay, if C6 is oxidized, okay, this is the sixth carbon. If C6 is oxidized, It will also result in formation of, yeah, it will also result in formation of an acid at the sixth place. And remember the name of this acid, it is uronic acid. The name of this acid, it is uronic acid. Okay, uronic acid. Now this acid, what is the use of this acid? Okay, since this structure it is of glucose, and this uronic acid, it is like no coming from the glucose, it is called, it is given a specific name beta, it is called as glucuronic acid. It is called as glucuronic acid. It is the name of this compound. When C6 carbon is oxidized, when C6 carbon is oxidized, okay, so it is coming from glucose, so that's why it is named as glucuronic acid. Okay, and what is the clinical significance beta? If we talk about the clinical significance, it helps in, okay, it helps in the conjugation of bilirubin. Okay, it helps in the conjugation of bilirubin. Okay, in much more classes beta we will be discussing each and everything, so as of now just mention the clinical significances. We will be discussing it like जैसे जैसे टॉपिक आगे बढ़ते जाएंगे, so you will be clear cut with each and every concept beta. So absolutely no need to worry. So it helps in conjugation of bilirubin. Okay, in physio you will learn it also again. And second point, it helps in the synthesis of glycosaminoglycans. This is our topic for today. I will be discussing this. Okay, glycosaminoglycans and also proteoglycans. Proteoglycans. So remember, beta, it is like the sugar attached with amino group. Okay, so we will be discussing glycosaminoglycans in detail, beta, after some time, right? So absolutely no need to worry. So what is the medical importance over here that we have to keep in mind? Glucuronic acid helps in the conjugation of bilirubin, and it helps in the synthesis of glycosaminoglycans or proteoglycans. That is our topic for today. So I hope you are very much aware when what will happen when C1 of glucose will oxidize, it will form gluconic acid, and when C6 is oxidized, it will form glucuronic acid. Be very clear with the names, beta. Be very clear with the names. Okay, so just give me a thumbs up if you are just like if you have understood what will happen in mild oxidation at C1 and C6, right? So yes, very good, beta. Very good. Raj Patel, very good. Very good, Kriti, very good. Very good, beta. Perfect. Harsh, perfect. 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 So yes, one more thing that I wanted to discuss, okay, before reduction, okay? Yeah, absolutely no need to worry. Yes, everyone is right. So let me just add one more slide, and I want to take one more concept over here. What will happen, beta? What will happen if both, okay? Now, take one more case beta, case number 3. If both C1 and C6 is oxidized, if both C1 and C6 is oxidized, so you already know that we have, for example, this is the glucose wala part, right? 1, 2, 3, 4, 5, 6. Okay, so you already know if both 1 and 6th carbon are oxidized, what will happen? Okay, there will be acid group at both the place C1 and C6. If they are oxidized, beta, okay, if they are oxidized, what will happen? COOH group, it will be at first position also, and it will be at sixth position also. And this structure where you can see the acid group at first and sixth both positions, okay, so that is something which is called as saccharic acid. Okay, this is called as saccharic acid. This is called as saccharic acid, beta. Okay, now this is the normal part. Now if I say, beta, in case of glucose, in case of specifically if I'm talking about glucose, If both carbon 1 and carbon 6 are oxidized, what will happen? It will be given a specific name. It is called as glucosaccharic acid. It is called as glucosaccharic acid. And remember, beta, in case of galactose, it is given a specific name that is called as galactosaccharic acid. Galactosaccharic acid. Okay, so yes, galactosaccharic acid, it is also called as mucic acid. Okay, so yes, mucic acid. Okay, so beta, again a clinical significance that you have to remember over here. Okay, this is given a specific test where we actually, yeah, it is called as the mucic acid test. Mucic acid test, beta. Okay, so remember, it is test for glucose, test for glucose or any other monosaccharide, beta, any other monosaccharide, galactose, yeah. So sorry, I'm writing test for glucose, it is test for galactose, yeah, it is test for galactose. And can you tell me a condition where galactose is intolerant? Is intolerant to the newborns. Can you tell me the condition, beta, where the galactose is intolerant to the newborns? I want everyone to answer this, as this is a very basic point that I have asked you. So yes, remember, it is test for glucose. Yes, anyone who would like to know. So this is seen in condition. It is called as galactosemia. It is called as galactosemia, beta. Okay, we will be discussing galactosemia. Okay, so no need to worry as of now. If you are like not aware of these points and test, we'll again come up with a, yeah, very good, Usman. It is the milk part that is not absorbed. Yes, it is galactosemia, beta. It is galactosemia. So you just have to remember as of now कि glucose का अगर पहला carbon oxidize होगा, so it is the acid group will be at, and it is called as gluconic acid. If 6 carbon, it is called as glucuronic acid. If both the carbon 1 and 6 is oxidized, it is saccharic acid. In case of glucose, glucosaccharic acid, and in case of galactose, galactosaccharic acid. Okay, and galactosaccharic acid, it is also called as mucic acid, and there is a test which is known as mucic acid test, which we do for glucose levels. Okay, and these are specifically done for the newborns like who are suffering from galactosemia. Yeah, so I hope this is clear, beta. Just give me a thumbs up in the chat box if this part is clear as of now. If this part is clear as of now, so चलो ये आगे और डिटेल में पढ़ते रहेंगे. So let us not, yeah, चलो. हर्ष, आशीष, वेरी गुड बेटा, वेरी गुड, वेरी गुड. जयश्री, वेरी गुड बेटा, वेरी गुड. जयनाम, वेरी गुड. चलिए आंसर देते रहिए. सेठी उस्मान, वेरी गुड बेटा, वेरी गुड, परफेक्ट, 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 परफेक्ट. यस, या, चलो. गैलेक्टोज है बेटा, मैंने ग्लूकोज लिख दिया है क्या? सॉरी, इट इज गैलेक्टोज. बार बार वो ग्लूकोज ग्लूकोज आ रहा है ना, तो इसलिए, या, इट इज टेस्ट फॉर गैलेक्टोज. टेस्ट फॉर गैलेक्टोज लेवल्स. लैक्टोज हो गया ना बेटा, आप खुद देखो, लैक्टोज इंटॉलरेंट हो जाएगा. पार्ट 1 and part 2 is there on the YouTube, beta. So now coming on to the reduction. Okay, now everyone should answer this question. Glucose on being reduced will form alcohol, right? It will form alcohol, and remember, it will form a compound that is called as sorbitol. Sorbitol is a type of alcohol only. Sorbitol is a type of alcohol only. No need to go in the structures, no one will ask you. So remember, the enzyme which is helping in the reduction process, it is called as aldose reductase. It is called as aldose reductase, and sorbitol, it is a type of alcohol only. Okay, and glucose is sugar. So when sugar is reduced, it will form alcohol, and when glucose is reduced, it is like sorbitol will be formed, and the enzyme which helps in this particular reaction, it is, yeah, aldose reductase. Okay, now where you will see this, okay, So there is something which is called as sorbitol pathway beta. Sorbitol pathway. Okay, sorbitol pathway. And remember that sorbitol pathway is actually a pathway that is taking place in our body which produces sorbitol. Okay, which produces sorbitol. Okay, so yes, we have, yeah. Now, this sorbitol is like seen in beta. Yeah, it is seen in diabetic cataract. Diabetic cataract, right? This sorbitol, it is seen in diabetic cataract. You already know the diabetic patients. Okay, the diabetic patients are more prone to suffer from cataract because the glucose is the higher levels of glucose will reduce it to like sorbitol, and obviously they will produce it will affect the eyes and it will not like, yeah, it will be a cataract which will be resulted from this production, right? So I hope this is clear to each one of you. I hope this is clear to each one of you. So yes, that is the first point that you have to remember in reduction. Yes, that is the first point that you have to remember in reduction. If galactose is reduced beta, if galactose is reduced, it will produce galact yes, हाँ क्लाउडी लेंस हो जाता है कैटरैक्ट तो तुम्हें पता है ओके बेटा ओके देयर आर वेरियस टाइप्स ऑफ कैटरैक्ट्स राइट डायबिटिक कैटरैक्ट इज जस्ट अ कॉम्प्लिकेशन ऑफ डायबिटीज ओनली right higher level of glucose can cause cataract okay and that is like due to this sorbitol when the glucose levels in the body are very high right so yes so it is galactosol right so yes or it is also known as dulicitol okay so yes now this compound beta it is having a clinical significance it is development of cataract in old age okay firstly you should note that it is like the cataract it will be seen in patients of galactosemia okay galactosemia you already know galactosemia right intolerant lactose yeah and obviously the galactose levels will rise and it will lead to the development of cataract in these particular patients okay so yes we have something called as mannose basically it will be reduced to form mannitol mannitol and remember beta mannitol what is the clinical significance it decreases intracranial tension now when I say intracranial tension you are aware of the fact whenever there is like yeah heavy bleeding right we give mannitol so that yeah it reduces the pressure okay and that is why this IV mannitol it is used in pharmacology as to decrease the intracranial tension right yes so yes now obviously fructose also if fructose is reduced if fructose is reduced beta it will give two alcohols okay it will give two alcohols either it can be sorbitol okay or it can be mannitol okay it can be sorbitol or either it can be mannitol okay so remember beta in reduction if glucose is reduced it is sorbitol okay sorbitol there is a pathway which takes place in our body which produces sorbitol it is called as sorbitol pathway and it is seen in diabetic it is responsible for the diabetic cataract galactose galactostol and it is responsible for the development of cataract in galactosemia remember mannose is reduced to mannitol mannitol is like used in pharmacological industry okay to decrease the intracranial tension we use IV intravenous mannitol okay fructose will again produce two types of alcohol sorbitol and mannitol okay Now I want every one of you, yeah, बेटा ये फार्मा में पढ़ोगे. आई कैन एक्सप्लेन द पाथवे ऑल्सो, बट बेटा इट्स बेटर इफ यू रीड इट इन फार्मा. धीरे धीरे सारी चीजें आती जाएंगी. लेट अस फर्स्ट कंप्लीट द टॉपिक एंड देन आई विल टेक अप योर डाउट्स एज वेल. सो लेट अस नाउ आंसर दिस पर्टिकुलर क्वेश्चन. व्हिच ऑफ द फॉलोइंग इज अ कीटोहेक्सोस. चलिए जल्दी जल्दी से बताइए, फटाफट फटाफट. कुछ बेसिक क्वेश्चंस कर लेते हैं फिर. हाँ, कुछ बेसिक क्वेश्चंस कर लेते हैं. यस. Kriti says C. Sanjay says fructose. Very good. Harsh C. Raj Patel C. Very good, beta. You already know glucose is an aldose sugar. Glucose is an aldose sugar. Ribose is aldose. Okay, aldopentose. Yes, fructose is ketohexose. Sethi Usman Chena Manchal. Very good, beta. Ketohexose. Galactose is aldohexose. Galactose is aldohexose. तो हमने पहले ही पढ़ा था कि नंबर ऑफ कार्बन एटम है, नंबर ऑफ कार्बन एटम है, एंड इट कैन बी लाइक 3 4 5 6, and we have aldehyde derivative that are called as aldoses, and we have ketosis. So yes, fructose is a ketohexose. Okay, it is having a keto group at second position. Okay, it is having a keto group at second position. Yes, everyone is right. Coming on to the next question. The branching point in glycogen has. चलिए बेटा जल्दी से आंसर दीजिए. The branching point in glycogen has. चलिए फटाफट से बताइए. The branching point in glycogen has. Okay, Sethi Usman, Jainam, Anshul. Yes, Kriti, very good beta. It is, yeah, it is the alpha 1,6 glycosidic linkage. मैंने कल भी बताया था कि ब्रांचिंग के टाइम. ओके, हर्ष, राजवर्धन, रिया, किरण, वेरी गुड. जयनाम, वेरी गुड बेटा. ब्रांचिंग के टाइम यू ऑलरेडी नो कि व्हेन द ग्लूकोज आर लाइक लिंक्ड टू ईच अदर इन द स्ट्रेट चेन. ओके, जब स्ट्रेट चेन में होता है इट इज अल्फा 1 to 4, बट व्हेनेवर इट इज linked in the form of branch, like it is alpha 1 to 6, right? Okay, perfect. Coming on to the next question. The oxidation of C1 and C6 of glucose produces. The oxidation of C1 and C6 of glucose produces. जल्दी से आंसर दीजिए बेटा फटाफट से. So I'm waiting for the answers. Everyone should answer this question. Yes. Sure. Kriti says D, Jainam D, Sanjay D, Medicare D, Raj Patel D. Okay, perfect. Rajvardhan, you already know. C1, yeah, C1 ऑक्सीडाइज होगा तो ग्लूकोनिक, C6 ऑक्सीडाइज होगा तो ग्लूकोनिक एसिड. ठीक है. अल्कोहल, sorry, glucose reduced होगा तो sorbitol. When C1 and C6 both are oxidized, it will give rise to glucosaccharic acid. Yes, everyone is right. Anshul, Harsh, very good. Coming on to the last question. Yeah. So which of the following statements about homopolysaccharides is true? Now beta, you are going to tell me inulin is made up of only fructose. चलिए आंसर दीजिए. Is this statement true? True or false बता दो. Is the first statement true? चलिए फटाफट से ट्रू और फॉल्स बता दो. Is the first statement true? चलो बताओ जल्दी से बताओ बेटा. Is the first statement true or not? Yes, it is true. Very good. Kriti, Raj Patel, MD, Harsh, Shraddha, very good. Very good. It is the true statement. Now, the second statement, dextran is a homopolysaccharide. Tell me, beta, it is true or not? Dextran is a homopolysaccharide. Tell me, it is true or not? चलो जल्दी से बता दो फटाफट से. Yes, it is true. Very good. Very good. It is true. It is true. Now, coming on to the third statement, mucopolysaccharides constitutes majority of the homoglycans. Constitutes majority of the homoglycans. Shraddha says C. Yes, beta, C is false. It is hetero. Yes, it is heteroglycans. Okay. Yes, heteropolymers are there. Yes, it is false. Cellulose is the major dietary fiber. Yes, it is true. It is true. Yes. ये पॉलीसेक्राइड्स होगा. ठीक है. सो यस, हेट्रोपॉलीसेक्राइड्स है बेटा. हेट्रोपॉलीसेक्राइड से होमो नहीं होता. सो आंसर इज थर्ड. आंसर इज थर्ड. Now coming on to the topic that we are going to discuss in detail, beta, that is glycosaminoglycans. Glycosaminoglycans. Now let us study, beta, what does this mean? Okay, what does this mean? Now if we just bifurcate the name, beta, GLY, okay, or I can simply say GLYCO, okay, so this refers to glycogen. Now this refers to glycogen, okay, and you already know glycogen is a carbohydrate. Glycogen is a carbohydrate, right? You already know the glycogen, it is a carbohydrate, right? Now okay, now better remember, when I say amino, okay, I'm talking about the amino sugar, okay. Glycans means sugar. So we have okay, now we have the sugar which is attached to the amino group, which is attached to the amino group. Okay, we have the sugar which is attached to the amino group. Now, this glycosa aminoglycans, okay, this glucosa aminoglycans, it is also known as mucopolysaccharides. Okay, it is also known as mucopolysaccharides. So in short, we also write it as MPS, mucopolysaccharides. Poly का मतलब तो समझ आता है सबको. Poly means many, saccharides means sugar. Many sugar units are there, and when I say muco, beta, muco means mucin. Okay, it was first like seen in mucin. Okay, these glycosa aminoglycans, that's why it is called as mucopolysaccharides. It is called as mucopolysaccharides. Okay, now you already know. Okay, tell me, beta, glycosa aminoglycans, it is homopolysaccharide or heteropolysaccharide? I have already told you this yesterday. तो फटाफट से आंसर बताओ, whether it is homopolysaccharide or heteropolysaccharide. जल्दी से आंसर बताओ इसका, whether it is homopolysaccharide or heteropolysaccharide. चलो, आई एम वेटिंग फॉर योर आंसर्स बेटा. जल्दी बता दो बेटा. यस कृति, वेरी गुड बेटा. इट इज heteropolysaccharides, right? It is a type of hetero. Very good beta. Sethi Usman, Raj, Harsh, Alfez, very good. Kiran, Ashish, very good. Rajvardhan, very good. Prince, very good beta. It is heteropolysaccharides. It's a type of heteropolysaccharides. So now beta, if we just try to, okay, conclude the definition based on this particular information, so you should remember beta, glycosa aminoglycans, okay, or I can simply say it as GAG, glycosa aminoglycans, okay. So GAG, I'm writing in short form beta, you should remember it is glycosa aminoglycan. It is a long, okay, it is a long unbranched, okay, it is a long unbranched, there is no branching beta, it is long unbranched heteropolysaccharide, heteropolysaccharide, okay, and it has a repeating disaccharide unit. It has a repeating disaccharide unit. Now when I say repeating disaccharide unit beta, you are very well aware with the fact that this is unbranched chain. So this is unbranched chain. Okay, this is a chain like this. It has no branches. It is a long chain. Okay, it is a long chain. And when I say heteropolysaccharide beta, it is composed of two sugar units. Okay, it is composed of two sugar units, right? So it is like this beta. Okay, so it is like this. So we have the repeating disaccharide unit. We have the repeating disaccharide unit. We have the repeating disaccharide unit. You can see it is repeating in the chain. Okay, it is repeating in the chain. Now, what is this repeating disaccharide unit, beta? Repeating disaccharide unit. So this repeating disaccharide unit, it is made up of two components. Okay. So remember, beta, the repeating disaccharide unit, it is made up of two components. It is made up of two components. Okay, it is made up of two components. Okay, we have amino sugar, we have amino sugar, and we have acidic sugar. Acidic sugar. We have amino sugar and we have acidic sugar. Now, can you tell me one acidic sugar? Okay, can you tell me the example of an acidic sugar, beta? We have just discussed over right now. Like, I want everyone to answer this question. We have just discussed acidic sugar from glucose. Okay, tell me. From glucose, which as like, yeah, it is called which acid, beta? जल्दी से आंसर दे, तो ये तो अभी बताया मैंने. Yes, we have gluconic acid. Very good. Very good, beta. It is acidic sugar only ना. देखो शुगर है जिसमें एसिड ग्रुप प्रेजेंट है. ऑब्वियस सी बात है. यस, ग्लूकोनिक एसिड. वेरी गुड. वेरी गुड. अभी जस्ट पढ़ाया तो है मैंने, राजवर्धन. वेरी गुड. वेरी गुड. नाउ अमीनो शुगर, बेटा. अमीनो शुगर, it is again classified into, like, we have glucosamine and galactosamine. Okay, means the sugar group, it is attached to the amino group. Yes, everyone is right. Divyanshi, very good. Rajvardhan, very good, beta. Very good. It is the gluconic acid. अगर हम अमीनो शुगर की बात करें, बेटा, शुगर which is attached to the amine group, तो ग्लूकोज अगर अमीनो ग्रुप से अटैच्ड है तो व्हाट विल बी द नेम? ग्लूकोसामीन. इफ गैलेक्टोस इज अ शुगर इज अटैच्ड टू अमीन ग्रुप, इट इज गैलेक्टोसामीन, right? And when we say acidic sugar, beta, you have already answered this. Yes, we can have many things. We can have glucuronic acid. Okay, yes, glucuronic acid we can have. Yes, many acid, glucuronic acid as well. And there is one more acid that you have to remember. It is iduronic acid. It is iduronic acid. Majority this acid sugar, it will be a part of this glycosaminoglycans. So yes, I hope this is clear. Okay, so I hope this is clear. When I say heteropolysaccharide, beta, remember the monomer unit can vary. The monomer unit can vary. Glucuronic acid, iduronic acid are the ones which are a part of this glycosaminoglycans. Just give me a thumbs up, beta, if you are, like, you understood the basic structure of glycosaminoglycans. कि किस किस चीजों से बना हुआ है. एक तो कार्बोहाइड्रेट हो गया, ठीक है. अमीनो से अमीन ग्रुप हो गया. अमीन ग्रुप दो तरह, अमीनो शुगर हो गया, ठीक है. कार्बोहाइड्रेट विद अमीनो ग्रुप, ग्लूकोसामीन या गैलेक्टोसामीन, और एक एसिडिक शुगर, ठीक है. सो यस. चलो, हर्ष, राज, जयनाम, प्रिंस, किरण, वेरी गुड बेटा. दिव्यांशी, वेरी गुड बेटा. So repeating disaccharide units, beta, you are very well aware. Yeah. So yes, Kiran, very good. चल बाकी भी आंसर दे दिया करो, बाकी भी बता दिया करो. Yeah. Now, coming on to the properties of glycosaminoglycans, beta. The first property that you have to remember, they are negatively charged. They are negatively charged. Now they are polyanions. Beta. They are negatively charged. Why they are negatively charged? Because of different sugar molecules. Okay, because of different sugar molecules, they are negatively charged, right? Okay. Now you already know when I say sugar, beta, Okay, when I say sugar, you already know we have two types of sugar. We have amino sugar and we have acidic sugar. We have acidic sugar. So yes, we have amino sugar and we have acidic sugar. Amino sugar, okay. Yes, amino sugar beta. Yes, amino sugar it is positive, but but this amino sugar, okay, it is attached to, okay, in the compound it is attached with sulfate ions, okay. It is also attached with, yeah, acetyl group CH3COO minus, okay. And acidic sugar beta you already know, acid कहने का मतलब है COOH or COO minus. So this minus beta it contributes to the negative charge of GAG. It contributes to the negative charge of GAG. Yes, I hope this part is clear beta. I hope this part is clear. चलिए बेटा फटाफट से बता दो, जल्दी से बता दो। चल ठीक है, ठीक ठीक है, ठीक है। Second point beta that you have to remember, that you have to remember, these GAG basically they repel each other. They repel each other. Now, when I say they repel each other, beta, you should know that the mucus secretions are slippery. The mucus secretions are slippery. Beta, whenever you take mucus on your fingers, beta, it is slippery. Now, mucus, जो fluid होता है ना, the mucus fluid, it is slippery in nature because it is rich in GAG. Okay, it is repelling each other. That's why these mucus secretions are slippery. Okay, so yes, they repel each other, and that's why they are like slippery in nature. Okay. Now, beta, as they are negatively charged, okay, what they will do, they absorb water. They absorb water because of negative charge. They absorb water because of negative charge. They absorb water because of negative charge. You already know water is like abundant in the extracellular matrix, right? So obviously if they are absorbing water, that is due to their negative charges. Okay, now whenever beta, okay, now the fourth point that I'm writing it over here, whenever it absorbs water beta, what will happen, this it will swell. Okay, okay, so remember beta, whenever when it absorbs water, what will happen beta, it will swell. Okay, it will swell, it is the basic nature, it will swell. Now when water is removed, when water is removed from glycosaminoglycans, what will happen, it will revert, it revert back to its original position, revert back to its original position. Just like you know a rubber band beta, when you stretch the rubber band and when you leave the no when you leave your hands, this rubber band will no take up its original shape and size. That is due to the property of elasticity, right? And that is something what is happening whenever GAGs absorb water, it will swell, and whenever whatever will be removed from GAG, what will happen, it will come back to its normal shape or position, right? And that is something which is called as compressibility of GAG. It is called as compressibility of GAG. Compressibility of GAG. Okay, it is called as compressibility, or we call it as resilient. Okay, we call it as resilient or resilient property of GAG. Resilient property, and because of this resilient nature of glycosaminoglycans beta, because of this resilient nature of glycosaminoglycans, it is seen in like cases of synovial fluids. Okay, yeah, synovial fluids, and you already know vitreous humor. Vitreous humor. Vitreous humor is seen in which organ of the body? Vitreous humor, it is seen in which organ of the body? Beta, kindly comment. I want to know if you know this or not. Where is the vitreous humor present in our body? चलो जल्दी से बता दो. Where is the vitreous humor present in our body? Yes, Kriti, very good, beta. It is the eye part. It is the eye part. Kiran, very good. Anshul, very good. Shraddha, very good. Sundari Ellen, very good. Sethi Usman, very good. Vitreous humor, eye में होता synovial fluids. Yeah, mostly at the joints you can see these synovial fluids. Okay, and these are, yes, that is why beta these glycosaminoglycans are like mostly, or yeah, these sites, when I say synovial fluid or vitreous humor, they are highly resilient. Why? Because of these properties of GHE. They are found it over there. Like, yes. So I hope, yes, everyone is right. I hope this particular properties of GHE are clear to each one of you. These properties are clear to each one of you, beta. Just give me a thumbs up in the chat box if it is clear to each one of you. Chalo. Chalo. Raj Patel, Kriti, very good, beta. Very good, very good. So some GAGs which are we going to discuss, Harsh, Jainam, Anshul, Surbeen, very good, beta. So the GAGs which are like our point of discussion for today, it is chondroitin sulfate. Okay, we are going to discuss about keratin sulfate, heparin sulfate, we have dermatin sulfate, we will be discussing this one by one, so no need to worry. We have heparin, and we have hyaluronic acid. Hyaluronic acid. So we have chondroitin sulfate, keratin, heparin, dermatin sulfate, heparin, and hyaluronic acid. So let us discuss one by one. Let us discuss one by one. So chondroitin sulfate, you have to remember that it is the most abundant. Okay, what are the medical points that are important over here? Most abundant glycosaminoglycan. Okay, it is the heterogeneous, most heterogeneous glycosaminoglycan. And remember, beta, chondro word, it is used for what? Chondro word, it is used for what? I want the answers in the chat box. Chondro word, it is used for what? I want everyone to answer this. बोन के लिए ऑस्टियो वर्ड यूज करते हैं. बोन के लिए वी यूजली, या तो कॉन्ड्रो वर्ड किसके लिए यूज होता है बेटा? यस, ऑब्वियसली बात है, कार्टिलेज के लिए होता है ना बेटा. कॉन्ड्रोसाइट्स, chondroblast. So this chondro is for cartilage only, right? Osteocytes, yes. Ridhanshi, yes. Chondrocytes, Usman, Priti, Raj Patel, very good, beta. So remember, beta, this chondroitin sulfate, it is seen in cartilages. It is seen in cartilages. That is again a takeaway point. It is seen in cartilages. No need to ratify, beta. Just remember the name, chondroitin sulfate. Okay. It is seen in cartilages. Also, this is a compound which can be seen in bones as well. Bones as well. Okay, it can be seen in bones as well. So what is the function of this chondroitin sulfate? It is responsible, beta. It is responsible for compressibility, of cartilage and weight bearing bone. Weight bearing bone. So just you have to remember it is the most abundant and heterogeneous GAG. Okay, it is seen in cartilages, it is seen in bones also, and remember it is responsible for compressibility of cartilage and weight bearing bones. So that is the like some points that you have to remember about this GAG. Now coming on to the keratin sulfate beta. Keratin sulfate. So keratin sulfate is so it is an acetyl group that is, it is N-acetyl glucosamine, N-acetyl glucosamine. So yes, N-acetyl glucosamine. Okay. So yes, it is an acetyl group. Okay. Yeah, it is an acetyl group, which is yeah attached to the amino sugar. Amino sugar क्या है? Glucosamine. Glucose भी sugar, amino भी sugar है, right? So remember that keratin sulfate beta, it is of two types. Keratin sulfate 1 and keratin sulfate 2. Okay, keratin sulfate 1 and keratin sulfate 2. Yes, keratin sulfate 1, it is isolated, or it is isolated from cornea. It is isolated from cornea. And keratin sulfate 2, it is isolated, okay, like it is isolated from the cartilage. Okay, it is isolated from the cartilage, okay, and specifically the loose connective tissue. So it is present in the loose connective tissue. Loose connective tissue, right? Loose connective tissue. Now what is if this keratin sulfate beta, it is present in cornea? Remember beta, it is responsible for corneal transparency. It is responsible for corneal transparency. It is responsible for transparent nature of the cornea. बेटा आपके आगे eyes में the outermost structure बेटा, it is the cornea only which is transparent, right? और इस transparency को maintain करने के लिए it is the keratin sulfate which is like keratin sulfate 1 which is responsible for the maintenance of this transparency. Now my question to you people is, आइडियली जो हमने स्ट्रक्चर पढ़ा था बेटा, आइडियली जो जीएजी का स्ट्रक्चर पढ़ा था, सो यू शुड रिमेंबर बेटा कि उसमें दोनों चीजें होनी चाहिए, अमीनो शुगर भी होना चाहिए और एसिडिक शुगर भी होना चाहिए. तो क्या इसमें अमीनो शुगर प्रेजेंट है? यस, अमीनो शुगर इज प्रेजेंट, बट व्हाट इज एब्सेंट बेटा ओवर हियर? What is absent over here? Tell me in the chat box what is absent over here. So that is a takeaway point that has been asked in the exam multiple number of times beta, that it is a yes, इसमें क्या absent है बेटा? Acidic sugar absent है ना? So I can simply say it is a GAG with no uronic acid. It is a GAG with no uronic acid or any acid. It is it is absence of this acid sugar which is like only absent over here, right? So it is N-acetyl galactosamine, right? So now coming on to dermatin sulfate. Now again the name suggests dermatin, so where it will be present beta? Where it will be present? I want the answers in the chat box beta. Use your brains and tell me dermatin sulfate, where it will be present. चलो दिव्यांशी बिल्कुल ठीक है, प्रिंस राज, किरण वेरी गुड, यश, श्रद्धा, किरण, संजय वेरी गुड बेटा. इट इज प्रेजेंट इन स्किन. ओके, इट इज मोस्ट abundant GAG in skin. It is the most abundant GAG in skin. It is the most abundant GAG in skin. Okay, it is the most abundant GAG in skin. Yes, Divyanshi, Jainam, Sithi, Usman, Jayashri, very good beta. And remember it is widely located GAG. It is present all over. So what is the largest organ of the body beta? It is widely located GAG. Meanwhile you just answer the question, which is the largest organ of the body? Which is the largest organ of the body? I want the answers from each one of you. Sure. It is the widely located GHE and it is the most abundant GHE in skin. Yes, beta, it is the skin only. Beta, liver is a gland. Again, it is skin, beta. Sanjay Usman, very good. It is the skin that is the largest organ of the body. Right? Keratin sulfate हो गया, chondroitin sulfate हो गया, dermatin sulfate हो गया, heparin sulfate आ जाओ. Heparin sulfate. So remember, beta, when I say heparin sulfate, so you should know it is made up of two things. Glucosamine. Now, when I say glucosamine, it is amino sugar. Okay, it is a disaccharide unit now. Amino sugar, and we have specific component over here, that is glucuronic acid. Glucuronic acid, and you already know glucuronic acid, it is acidic sugar. So it consists of amino sugar and acidic sugar both. Yes, that is the composition of GAG. Yes. हाँ ठीक है, कोई बात नहीं बेटा, नहीं पता अभी पता चल जाएगा, तो कोई दिक्कत नहीं है, ठीक है. So yes, now where this heparin sulfate is seen, that is again an important question. It is seen in plasma membrane receptors. Okay, it is seen in plasma membrane receptors. It is seen in plasma membrane receptors. Okay, now you already know that plasma membrane has certain receptors, and these receptors are like, yeah, it is seen, this heparin sulfate is seen over there. For example, if you want, you already know an enzyme that is called as lipo का मतलब होता है lipid protein, lipoprotein lipase. Okay, lipoprotein lipase is anchored by heparin sulfate. बेटा anchored का मतलब होता है सपोर्ट करना. This particular enzyme, when I say ACE, beta, ACE is an enzyme. So this enzyme is supported by, the function of this enzyme, it is supported by heparin sulfate. Okay. Now again, if you want to know, you already know the basement membrane. The basement membrane of renal glomeruli. Okay, what will be there? Heparin sulfate will be present. Heparin sulfate is present. Okay, now you already know the renal glomeruli have negative charge. So renal glomeruli has negative charge. तो renal का जो glomerulus है उस पे negative charge होता है बेटा, and that negative charge, it is actually coming from where? Heparin sulfate. I have already told you that comes from heparin sulfate. So you should remember beta that heparin sulfate, it is responsible for charge selectiveness. Okay, it is responsible for charge selectiveness of renal glomeruli. It is responsible for charge selectiveness of renal glomeruli. You already know beta. Let us just understand with the help of an example. You already know albumin. Now, why albumin cannot cross this glomerulus wala part? Why it cannot cross this glomerulus wala part? Because remember beta, albumin का भी कौन सा चार्ज होता है? नेगेटिव चार्ज होता है. नेगेटिव चार्ज होता है. और जो ये बेसमेंट मेम्ब्रेन है, ये जो बेसमेंट मेम्ब्रेन है रीनल ग्लोमेरुलस की, this is also composed of negative charge which is coming from heparin sulfate, so obviously albumin का भी negative charge, heparin sulfate का भी negative charge that is present in the basement membrane, they will repel each other. That's why the albumin, okay, albumin, it is not coming in the GFR, glomerular filtrate. Okay, so remember that this heparin sulfate, it is present in synaptic vesicles also. Okay, These are present in synaptic vesicles also. Okay, it is present in synaptic vesicles. So that are all the takeaway points, beta. Very, very important slide. So I will just try to put a dollar sign over here. So already you should remember the component, the biochemical components, okay, the amino sugar and the acetic sugar. It is seen in plasma membrane receptors. Over the plasma membrane, there are several receptors where it is present. Even the enzyme, that is lipoprotein lipase, the function, it is anchored, it is supported by heparan sulfate and the basement membrane of the glomerulus. Okay, they contain this heparan sulfate, or basement membrane में negative charge आ जाता है because of this heparan sulfate. Even the molecules such as albumin will not be able to cross this basement membrane of renal glomerulus, and that is why of the so reason that these both negative charges will repel each other. So that is responsible for charge selectiveness of renal glomeruli. I hope this is clear to each and everyone, beta. Just give me a thumbs up that heparin sulfate, it is clear to each and everyone over here. चलिए फटाफट से आंसर दे दीजिए if it is clear to each and everyone. Yeah. Priti says yes sir, it is clear. Raj, Anshul, Harsh, very good beta. Kiran, Rajvardhan, very good, very good, very good beta. Jainam, very good, very good. Riya Singh, very good, very good beta. Shraddha, very good. दिव्यांशी, perfect, perfect, perfect. चलिए सेठी उस्मान, वेरी गुड. चल बेटा, डर्मेटिन हो गया, केराटिन हो गया, सब कुछ नाम से याद रखिएगा. ओके, कॉन्ड्रोइटिन सल्फेट, तो ये सारी चीजें तो हमने पढ़ ली. अब आ जाओ हाइलुरोनिक एसिड पे. हाइलुरोनिक एसिड पे. सो यस, usually beta, the sulfate groups are attached, but over here what is the constituent beta? It is N acetyl glucosamine plus glucuronic acid, plus glucuronic acid. Now बेटा, you already know कि ये जो अमीनो शुगर है, अमीनो शुगर है, वो यूजुअली अटैच्ड होता है दिस अमीनो, okay, this amino sugar. I will just explain it. This amino sugar beta, it is positive in nature and it attaches to sulfate. Okay, it attaches to sulfate, but over here it is not attached to sulfate, it is attached to acetyl group. It is attached to acetyl group. So remember, it is in hyaluronic acid, there is no sulfate group, no sulfate group. Okay, so yes, it is not covalently attached to proteins, not covalently attached to proteins. Yes, not covalently attached to proteins. Okay, so yes. Now what is the function of this? Then it helps in the cell migration. Cell migration. And yes, when I say cell migration beta, so the very first thing that comes into my mind, it is tumor cell migration. Tumor cell migration, and that is something called as metastasis. Metastasis. Okay, from one region the cancer cells are spreading into some other region. For example, मुझे cancer हो गया stomach में and it is spreading to the lungs. Okay, so that cells, that tumor cells will migrate from one organ to another, and that is something called as tumor cell migration, or that is metastasis. एक होता है benign cancer. Benign का मतलब होता है it is located at one place. और एक होता है metastatic cancer. It can spread from one organ to another. Okay, और ट्यूमर सेल की मेटास्टेसिस में और मूवमेंट फ्रॉम वन ऑर्गन टू अनदर, दैट इज डन बाय hyaluronic acid. That is done by hyaluronic acid. Okay, also it plays a role in morphogenesis. जेनेसिस का मतलब होता है फॉर्मेशन, मॉर्फो का मतलब एक्सटर्नल. ओके, एक्सटर्नली जो या during the development or during the morphogenesis, okay, of the embryo. Yes, it is. And also this hyaluronic acid plays an important role in wound repair. It plays an important role in wound repair. Yes, After that, hyaluronic acid, it is also found in many bacteria. Okay. Now coming on to heparin. Now can anyone tell me about heparin, beta? Heparin के बारे में बता दो. Okay. What is it? What does it do, बेटा? इसकी बेसिक प्रॉपर्टी अगर फिजियो से थोड़ी बहुत याद है तो बता दो. वैसे तो इतना कुछ है नहीं इसमें. So yes, it is an anticoagulant, बेटा. It is an anticoagulant. एंटीकोगुलेंट का मतलब क्या होता है? चलो यही बता दो फटाफट से चैट बॉक्स के अंदर. इट इज एन एंटीकोगुलेंट. व्हाट इज मेंट बाय एंटीकोगुलेंट, बेटा? एंटीकोगुलेंट का मतलब बता दो. एंटीकोगुलेंट का मतलब बताओ. उस्मान ठीक है, एंटीकोगुलेंट है. हेपारिन एंटीकोगुलेंट है. हाँ, मेड अप ऑफ ग्लूकोसामीन. हाँ, एंटीकोगुलेंट ब्रेक डाउन क्लॉट, स्टॉप ब्लड क्लॉटिंग. ब्लीडिंग, स्टॉप ब्लीडिंग, प्रिवेंट्स मेक क्लॉट. बहुत बढ़िया. मेड अप ऑफ ग्लूकोसामीन एंड आइड्यूरोनिक एसिड. Hyaluronic acid. Okay. Remember, beta, heparin, it is only present in the cells. Okay. Only intracellular. जब बेटा मैं इंट्रासेलुलर बोलता हूं तो सेल के अंदर की बात कर रहा हूं. इंट्रा मींस इनसाइड द सेल. या इट प्रिवेंट्स द ब्लड क्लॉट्स. ओके. So I'm talking about the intracellular GAGs. Okay, it is present inside the cell. Okay. And remember, beta, intracellular, which cells? Actually, it is the mast cells. Okay, it is the mast cells, which you can see in, yeah, even in the lungs also you can see it over here. And there are many much more places where you can find this, right? So yes. Coming on to the next part, that is the proteoglycans. Beta. So remember, beta, again the name suggests proteo means protein and glycans means GAGs. Now it simply indicates that GAGs which are attached to proteins. Okay, so proteoglycans will contain 5% protein and 95% GAGs. Okay, GAGs. So obviously we are talking about glycoproteins over here. Glycoproteins. And remember, when I say glycoproteins, beta, it is carbohydrate plus protein. Okay, carbohydrate plus protein. Now this protein, it is in the form of glycoproteins. Now over here, yeah, you should remember it is like carbohydrates plus protein. Now how a proteoglycan is formed? So you should know, beta, if suppose this is a protein. Okay, suppose this is a protein. If suppose this is a structure of the protein. Okay, and yeah, what is happening, beta? The GAGs are attached to these proteins. Okay, the GAGs are attached to this proteins. So yes. So these are GAGs which are attached to these proteins. Okay, GAGs are attached to proteins. And they are attached to proteins with a stalk. Okay, you can see a stalk over here. They are attached to proteins with a stalk. Okay, and that is why it is given a name as bottle brush shape. It is given a name as bottle brush shape. Okay, shape of proteoglycan monomer, it is bottle brush shape. So I hope this is clear to each and everyone over here. So in proteoglycans, you just have to remember it is composed of proteins and GAGs, and the content of protein, it is 5%, and GAGs, it is 95%. Right? I hope this is clear to each one of you over here. Just give me a thumbs up in the chat box, beta. Just give me a thumbs up. Proteoglycans, heparin, hyaluronic acid, heparin sulfate, dermatan sulfate, keratin sulfate, chondroitin sulfate, all the properties of GAG, glycosaminoglycans, oxidation and reduction, it is clear, along with the questions that we have discussed. Raj, Harsh, very good. Kiran, Jainam, very good. Prince, very good, beta. Very good. Very good. Now, coming on to the questions. Okay, first two questions I will discuss, and the other questions you guys are going to answer me. I want to see how many of you are like well prepared. So, yes, सिंथेसिस ऑफ ग्लाइकोसिंथेसिस का मतलब होता है बेटा बनना. ग्लाइकोसा अमीनो ग्लाइकेंस जो है वो कहाँ बनते हैं? रिमेंबर बेटा, इट इज एंडोप्लाज्मिक रेटिकुलम. अगेन आई एम सेइंग सिंथेसिस बेटा, एंडोप्लाज्मिक रेटिकुलम and Golgi apparatus. Endoplasmic reticulum and Golgi apparatus. Endoplasmic reticulum and Golgi apparatus. Okay. Degradation of GAG. Degradation मतलब GAG कहाँ पे नष्ट हो जाते हैं? Basically they are destroyed in lysosome. They are destroyed in lysosomes. अगर बेटा if there is a defect, if there is a defect in degradation, remember beta, that constitutes a group of disorder that is called as mucopolysaccharidosis. Saccharidosis. I will be taking this topic tomorrow beta. All the diseases I will discuss tomorrow. Mucopolysaccharidosis, that is also called as MPS. Mucopolysaccharidosis. वो म्यूकोपॉलीसैक्राइड्स था, इट इज म्यूकोपॉलीसैक्राइडोसिस. अगर डिफेक्ट होता है, डिग्रेडेशन नहीं हो रहा है लाइसोसोम्स में, if there is no degradation which is happening in the lysosomes, if there is a defect in the degradation process, it will result in a group of disorders. We have a group of disorders that is called as mucopolysaccharidosis, and I will be taking up these disorders tomorrow. Okay, I will now start with the clinical part beta, which is relevant. Now the third question beta, I want every one of you to answer. GAG with no uronic acid. चलो जल्दी से बता दो. GAG with no uronic acid. कृति very good बेटा. बाकी सब भी आंसर बताओ. GAG with no uronic acid. Yes, Jainam very good. Kriti very good. Harsh, Shraddha very good बेटा. Raj Patel, Kiran very good बेटा. It is the keratin sulfate बेटा. It is the keratin sulfate. It is the keratin sulfate. Now GAG with no sulfate group. Yes, Rajvardhan, Sethi Usman very good. GAG with no sulfate group beta. जल्दी से बताओ. No sulfate group. It is hyaluronic acid. These are all questions that has been asked in the exams. So GAG with no sulfate group, it is hyaluronic acid. It is hyaluronic acid. It is hyaluronic acid. Shraddha, Rajvardhan, very good beta. Now GAG which is an anticoagulant beta. GAG which is an anticoagulant. Yes, Kiran, हाँ नहीं वो वाला. कृति, श्रद्धा, संजय, प्रिंस बेटा हाइलुरोनिक नहीं है ना, हेपेरिन होता है एंटीकोगुलेंट ना. जैना अभी तो बताया था. हाँ सॉरी प्रिंस, अभी तो बताया था बेटा. किरण, उस्मान very good. Yes, it is heparin beta. GAG which is an anticoagulant, that is heparin. Most abundant GAG. Most abundant GAG. चलो जल्दी बताओ, what is the most abundant GAG? Yes, chondroitin sulfate. Very good, Harsh. Like I will ignore all the spelling errors because they are not required as of now. I just want your brains to work active. Jayashree, Kriti, Devyanshi, Shraddha, Raj Patel, Usman. Very good, Pita. It is the chondroitin sulfate. It is the chondroitin sulfate. Now next question. Shape of proteoglycan बेटा. अभी बताया मैंने shape of proteoglycan. चलो जल्दी से बता दो, what is the shape of proteoglycan monomer? It is, yes, Kriti, very good beta. It is the bottle brush shape. Sanjay, Shraddha, very good beta. Very good. It is the bottle brush shape. It is the bottle brush shape. It is the bottle brush shape. Okay. Now, the GAG that helps in cell migration. Yes, Kriti, very good. Shraddha, very good. Sanjay, very good, beta. It is hyaluronic acid. Raj Patel, very good, beta. It is hyaluronic acid. It is hyaluronic acid. Now, coming on to the next one, Kiran Prince, very good, beta. Sethi Usman, very good. Coming on to the next question, GAG that have a role in compressibility of cartilage in weight bearing. GAG that have a role in compressibility of cartilage in weight bearing. Kriti says chondroitin sulfate, Usman chondroitin, Jainam chondroitin, very good, beta. It is chondroitin sulfate. It is chondroitin sulfate. Now, GAG that plays a role in corneal transparency beta. GAG that plays a role in corneal transparency beta. चलिए कृति खंडेलवाल keratin sulfate ठीक है, but which keratin sulfate? I have told you there are two types of keratin sulfate, KS1 and KS2. हाँ प्रिंस जयनाम ठीक है, keratin वाला point ठीक है, but which keratin बेटा? Which keratin? हाँ we have the keratin sulfate 1. Yes, Kriti Raj Patel Harsh, very good. It is keratin sulfate 1. It is the keratin sulfate 1. Yes, Sanjay Jainam, very good, beta. Very good. Keratin sulfate 1. Coming on to the next question. GAG that determines charge selectiveness of renal glomerular membrane. GAG that determines charge selectiveness of renal glomerular membrane. राजवर्धन किरण प्रिंस सबका सही था. चलो इसका आंसर बताओ. Yes, that determines charge selectiveness of renal glomerular membrane. फटाफट से आंसर दो. Yes, Kriti, it is heparin sulfate beta. It is heparin sulfate. Very good, Harsh. Very good, Prince. Very good. Very good, beta. It is the heparin sulfate. It is the heparin sulfate. Kiran, very good. Kiran Yadav, very good, beta. इंट्रासेलुलर जीएजी. अब बताओ सेल के अंदर कौन सा जीएजी प्रेजेंट होता है? विच जीएजी इज प्रेजेंट इनसाइड द सेल बेटा? विच जीएजी इज प्रेजेंट इनसाइड द सेल? यस कृति, वेरी गुड बेटा. बाकी सब भी आंसर्स तो इज कृति राइट? या हेपारिन, हर्ष हेपारिन, राज हेपारिन. यस बेटा. श्रद्धा, किरण, वेरी गुड बेटा. इट इज द हेपारिन which is intracellular GAG. It is the heparin which is intracellular GAG. Okay, GAG in synaptic and other vesicles. Which GAG is present in synaptic? Synapse होता है ना, synaptic vesicles होते हैं. Which GAG is present in synaptic and other vesicles? Yes, Kriti, it is heparin sulfate. Usman, very good. नहीं उस्मान, dermatin कैसे होगा बेटा? Dermatin वाला तो dermatin sulfate हो गया ना. हाँ heparin sulfate होगा ना, which is in synaptic and other vesicles. It is heparin sulfate बेटा. Heparin है, dermatin नहीं है. It is heparin sulfate. Heparin sulfate. Now next question. Only GAG not, ध्यान से पढ़ना, not covalently attached to a core protein. Only GAG not covalently attached to a core protein. Yes, Kriti says hyaluronic acid. Beta, it is hyaluronic acid. Right? It is Harsh, very good beta. It is hyaluronic acid. It is only GAG. It is not covalently attached to a core protein. Okay. Now GAG which binds to low density lipoprotein, that you have to remember beta. It is dermatin. एक तो क्वेश्चन बाहर से भी. It is dermatin sulfate. Okay. It is dermatin sulfate. It binds to low density lipoprotein, lipid anchored with protein, lipoprotein, lipid anchored with protein. Surely, I hope all the questions are clear, beta. All the one-liner questions that I have discussed. Okay, all the topics that we have like taken up today, we have taken the oxidation of sugars. I've already told you about the oxidation. What will happen if C1, C6, if both C1 and C6 is oxidized? Reduction. Yes, reduction. The questions, the basic questions that I've discussed. Yeah. After that, glycosaminoglycans, the formation, the constituents, right? The properties, okay? After that, the major GAGs that we have discussed: chondroitin sulfate, keratin sulfate, heparin sulfate, dermatin sulfate, heparin, and hyaluronic acid. After that, we have already discussed this in detail, guys. We have discussed proteoglycans, and we have discussed the synthesis and degradation of GAG also, and all the important one-liners that are like important for exam point of view. So I hope today's lecture is clear to each and everyone. Beta, just give me a thumbs up in the chat box. Just give me a thumbs up in the chat box if today's lecture is clear to each one of you, beta. Chali, just give me a thumbs up in the chat box if today's lecture is clear to each one of you. Chal. Raj Patel, very good. Sethi Usman, very good. Kriti, very good. Beta, tomorrow we'll start with something clinical. Beta, tomorrow I will talk about the diseases. Okay. अगर GHEs degrade नहीं हुए lysosomes में तो क्या होगा? It can result in which diseases? So we will be talking about these tomorrow, and we will see the cases. Okay, so yes, tomorrow's lecture will be highly conceptual, beta. That is something I will take कि मतलब अगर चार जितने lectures हुए हैं सबसे important वाला lecture क्या है, तो आज और कल वाला lecture होगा. Yes, beta, notes will be shared after all the topics, after the basics of this biochemistry is done. I will start with the carbohydrate metabolism, but before that, like I want to complete with the basics by tomorrow. Yeah, then we will start with glucose and other thing. Yes, I hope it's like way clear to everyone. Yes, so it will be shared after like the whole biochemistry of carbohydrates is done. So yes, after one or two lectures maybe, as what sir told me to convey you people. So कोई doubt तो नहीं है बेटा? No doubts I suppose. No doubts I suppose बेटा. चलिए. बाय बाय, खुश रहिए. बाय.